Hello everyone, welcome to ITLS Academy. The topic of today's lecture is our ongoing topic, that is rebuilding the nervous system with the help of stem cells. We have covered a brief introduction in part 1 and we have done some detailed study also. Some things was left we will study in this video. So let us start. Fetal tissue transplants in Parkinson's disease research. First point is the idea of growing dopamine cells in the laboratory to treat Parkinson's disease is the most recent step in the long history of cell or tissue transplantation. The concept was and still is straightforward. That is, implant cells into the brain that can replace the lost dopamine releasing neurons. Next point is fully developed and differentiated dopamine neurons do not survive transplantation. Full functional recovery depends on more than cell survival and dopamine release. Surgical approach involved the transplantation of dopamine producing cells found in the adrenal glands which sit atop the kidneys in the abdomen. Next point. Surgeons in the United States, however, observed only very modest and inconsistent improvement in their patients' symptoms, and any gain disappeared within a year after surgery. Furthermore, it became clear that the risk associated with the procedure, which required both brain and abdominal surgery on patients who are often frail and elderly, outweighed the benefits. Another strategy based on transplanting developing dopamine neurons from fetal brain tissue has fared better. By the early 1980s, Anders Zockland and others had shown that transplantation of fetal tissue into the damaged area of the brains of rats and monkeys used as models of the disease could reverse their Parkinson's-like symptoms. Subsequently, researchers refined their surgical techniques and showed that functional recovery depends on the implanted neurons growing and making functional connections at the appropriate brain locations, essentially finishing their maturation by integrating into the adult host brain. A major weakness in these initial studies was that they were all done open label, meaning that both researchers and patients knew which patient received the transplanted tissue. So, this was a major weakness. When appropriate, the best test of a new therapy is a placebo controlled double blind trial in which neither researcher nor patient knows who has received the experimental treatment. And this was a major step as neither researcher nor patient knows about the transplant. Although in the initial studies this weakness was persist, but finally researchers has developed a method by which neither researcher nor patient knows the experimental treatment. This is an image showing this is an image of positron emission tomography, in short PET. This is PET image from Parkinson's patient, and this is before and after fetal tissue transplantation. You can understand the differences that you can see here differences are showing. This is before surgery and this is after surgery. After surgery image have a lower type of disease symptoms. You can see here symptoms are reduced. Just compare these two images. You will find that this black portion decreased in the after surgery image. Here transplantation of the dopamine neuron has occurred. The image that is taken before surgery, this is left side image. This is before surgery image and it shows uptake of a radioactive form of dopamine. Dopamine is a red form, you can see here. This is dopamine, red form. And uptake of the dopamine is only in the caudate nucleus. And it indicates that dopamine neurons have degenerated. You can see here this has very less amount. If we talk about after surgery image, you can see here dopamine concentration has increased. This image is taken after the 12th month after surgery, and this image is from the same patient, and it reveals increased dopamine function and especially in the putamen. While in this image this was only in the caudate nucleus, but this image have dopamine concentration in caudate nucleus as well as in putamen. In the mid 1990s, NIH approved funding for two rigorous clinical trials. There were two clinical trials, we will see both of them. These clinical trials are of fetal tissue transplantation for Parkinson's patients. Both studies provided for placebo control. The results of one of these trials, led by Kurt Fried, were published recently. Compared with control, patients who received the fetal tissue transplant showed no significant benefit in a subjective assessment of the patient's quality of life, which was the study's primary endpoint. Moreover, 2 years after surgery, 5 out of 33 treated patients developed persistent dyskinesia, uncontrolled flailing movements. These flailing movements had not been observed in the open label work described above. So, the Fried study results nonetheless provide important information about the ability of dopamine neurons to survive in humans. Moreover, PET scanning data, I am moving to that image. This is PET scanning data as well as autopsies of two patients who died of unrelated causes several months after the surgery showed that many of the dopamine neurons survived and grew. Raising neurons for replacement in patients with Parkinson's disease. What Parkinson's researchers ultimately want is a renewable source of cells that can differentiate into functional dopamine neurons. This is a major point. What they wanted when placed in the stratum. Laboratory grown cells derived from a stem cell may be the best potential alternative source for transplantable material. Whether the cells ultimately implanted are half differentiated or completely immature, however, researchers need a reliable source. To that end, they have identified a whole host of different immature cells that may have the potential to become, among other things, that is, dopamine neurons, and they are now in the process of sorting out how best to make them do so. Neural stem cells isolated from animals and humans cannot be grown efficiently in the lab without changing them in some way, such as by engineering them to express a gene normally turned on only early in development. Researchers are making rapid progress that they were able to expand a population of neurons from embryonic mouse brain in culture. Turning on the brain's own stem cell as a repair mechanism. Here, this is really a turning point in the brain repair. Let us see what points are given here. First point. Stem cells in the adult to primate brain occurs in two locations. What they are? One, the subventricular zone. This ventricular area under the other one details of hippocampus. All the positions detailed. If you watch, then I recommend you to watch them first. So these are two locations in which stem cells are found. Researchers showed in the mid 1990s that when the brain is injured, stem cells in these two areas proliferate and migrate towards the site of the damage. The researchers are now trying to discover how far is the kind of response can go toward ameliorating certain kind of damage. Next point. Recent research shows the direction that this may be heading for Parkinson's disease. Another point is brain's normal repair process. What was the normal repair process? Let me recall that there were hematopoietic stem cell and embryonic cells. Embryonic stem cells. These cells were used in the brain repair process. We have discussed in detail all these points, how these are involved in the repairing of brain. So, for the brain's normal repair process may never be adequately triggered in a slowly developing degenerative disease like Parkinson's, and that providing more TGF can turn it on. TGF injected into healthy rat brain causes stem cells in the subventricular zone to proliferate for several days, after which they disappear. Let me draw to understand you. This is a rat. Although this rat is very funny, this is looking like a bird. This is a rat. TGF injected into healthy rat brain. If this is a brain, we have injected TGF This is a type of growth factor that is injected in a healthy rat's brain. What this causes? This helped the stem cells that were present in the subventricular zone, as we have seen that stem cells are found in two locations. First one was subventricular zone and next one was ventricles. So this TGF, this growth factor, causes the stem cells in the subventricular zone, in this zone, to proliferate for several days. The stem cells of subventricular zone. These stem cells proliferated for several days. So this was an experiment, and the result of this experiment was that we can make proliferate stem cells of the subventricular zone by injecting with TGF. This TGF results in the increased life of the subventricular zone stem cells. These stem cells start divide, and these can form a lot of kind of further stem cells. We have seen various kind of repair processes. Let us move to the stem cells future role in spinal cord injury repair. As we know that spinal cord is also a part of brain. This is spinal cord connected to the brain, and we are going to see future role of stem cells in this spinal cord injury if it happens at some time or at some stage of life. So let us see. First point given here is complete restoration after severe spinal cord injury, for example, is probably far in the future. It is very difficult, and if it can ever be done at all. Next point is spinal cord injury patients would benefit greatly from an even limited restoration of lost functions. If there is limited restoration of functions, then it could work properly. Such limited restoration of parts of a patient's lost function is for some less severe types of injury, perhaps a more achievable goal. Another point given here is researchers have recently made the first step in learning to replace lost myelin producing cells. What is myelin? I hope you all remember what is myelin. We talk about a neuron. This is cell body. These are dendrites. This cell body have nucleus. This is the axon part of the neuron. These are synapse and synaptic lobes. This axon part is surrounded by some fatty sheath. This sheath is known as myelin sheath. This is the covering of neuron or axon part of the neuron. As we know that axon is an conducting part. So scientists learned how we can recover these myelin producing cells. If there is formation of an axon that is non-myelinated or that is not covered with this myelin sheath, then how can we restore these cells so that a kind of cells that can make this myelin sheath? So we can say scientists have succeeded in that kind of myelin producing cells replacement. Researchers have shown that stem cells can aid remyelination in rodents. As we know, all the experiment most of the times are performed in rodents. So this therapy was also discovered in rodents and it was successful as this remyelinated the rodent axons. Specifically, they found that injection of oligodendrocytes derived from mouse embryonic stem cells could remyelinate axons in chemically demyelinated rat spinal cord. This was a very huge discovery. Spinal injury researchers emphasize that much more basic and preclinical research must be done before attempting human trials. All things are still limited to rodents or rats. Still, human trials are not done. Although human trials are going on, but it is not a final step that we can directly transplant these cells in humans. So there are a lot of research that is to go to attempt human trials using stem cell therapies to repair the damaged nervous system. Whenever it will happen, it will be a huge step for the mankind. So thank you very much guys for watching this session. Follow ITLS Academy on various social media platforms like Instagram, Facebook, Twitter, YouTube, WhatsApp and LinkedIn. Also subscribe our YouTube channel ITLS Academy and do not forget to press the bell icon so that you can get notified whenever there is an upload to the channel. You can also get it on Google Play Store. Thank you very much for watching. Like, share and subscribe please. Thank you very much again.